been locked on and off when we haven't had undergraduates helping us there, but I'm glad to see everybody made it. Let me announce some upcoming seminars, and then I'll turn it over to Eileen to introduce Nikki today. The Energy Resource Group Colloquium is today from 4 to 5:30 in Giannini Hall. EHAVIORAL is speaking on examining fair access to reliable electricity through energy justice. IB seminar tomorrow is our own Charles Marshall, and the title of his talk is Beyond Darwin's Warm Little Pond, Macroevolution, Organismal Biology, and Origin of Life Scenarios. It's a small topic.
Come early for treats.
And come early for treats. Thank you, Annalise. That's tomorrow at 12:30 downstairs. Essig brunch Friday at 10 in 5053 in this building. Tyler Douglas is speaking. So Tyler, are you here? Your title is Modeling the Evolution of Chemical Defense in Drosophila melanogaster. Do you want to say anything else?
Yes, we're going over some early results from our chemical project, so hopefully we'll be interested.
Very good. Herbarium talk, botany lunch Friday at noon, 1002 DLSB, so downstairs. Christian Henry, who's a postdoc at UCLA and here at Berkeley, is talking about evolutionary climate relationships of leaf traits across the California and Hawaiian flora. Herp group, Monday, October 10th, so a week from Monday, 6:30. Ben Karen speaking. Ben, are you here? Okay. Sulawesi surplus of sphenomorphine skinks. Travelogue from Sulawesi and Borneo, fieldwork and latest results.
And just add, I'm planning to host it at my house, so y'all are invited.
Nice, very good. Next MVZ lunch. So next week we don't have MVZ lunch. We're having a faculty and staff meeting here at that time. And then Wednesday, so 2 weeks from today, Umalela is speaking. So Umi, are you here? Yeah. You want to say anything?
Just wait and see.
The title I have is Living in Fast Flowing Streams. Colon, blah, 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 blah. I'll save the rest for later. Are there any announcements? Is Jim still on? He hasn't gotten it out. What's
Still on
All right.
the 8th, maybe. We've heard nothing.
He told me he'd be back for her group, so.
And Rory just got back from the field, you know, supposedly collecting birds. I don't know. He went to some tropical island. We have to get an update on field work for all these people doing interesting field work. All right, I'll turn it over to Eileen, who will introduce.
All right. Well, it's my pleasure to introduce Nikki Lee, who's a new postdoc here in my lab. Nikki did her undergraduate work at Cornell University, where she majored in both biology and English. So I have to ask, did you hang out in Temple of Zeus or the Alfalfa work?
Temple of Zeus.
All right, a little bit of Cornell trivia there, but for those who know, yeah, big difference. She then went on to do her graduate work at the University of Massachusetts, where she worked under Annalise BEERY, so kind of coming full circle here. And then accepted a 1 year, at least temporary position as a special faculty diversity postdoctoral fellow at Colgate. So spent a year teaching, primarily it was a teaching position, before joining us here as an NSF postdoctoral fellow. So she's now back to doing research for a few years and kind of maybe trying to decide how to balance those two interests of hers. In terms of research, Nikki comes already highly accomplished. She's published 7 papers. She's first author on 4 of them, and they're in top tier professional journals like Hormones and Behavior and Frontiers in Behavioral Neuroscience. So clearly off to a strong start there, but she's also a very dedicated teacher who won awards at Massachusetts for her teaching, and that has translated into some of her postdoctoral work as well. What else did I want to say? There's a little note here, midterm practice key. I don't think that's relevant. But something else related to class. I'm particularly excited because this is an opportunity for my group to go in a new direction that, I study field behavior, trap rodents, things like that. And with Nikki's help, we're trying to launch into kind of a new direction, looking at some neuroendocrinological things in tuco. So I'm really excited. I have a few fun facts here. First is that I didn't realize this. I'm stepping away. Nikki has a black belt in Taekwondo, so I'm going to make sure I treat her well.
It's been like 10 years. I really,
Still. And she's also apparently a Jane Austen fan who's been on a Jane Austen pilgrimage
Yeah.
to the UK to travel visit some of the.
it was the closest thing to a spiritual experience. I just really liked it.
But again, maybe that's channeling that dual
Yeah.
undergraduate approach to biology and English, right? And so she obviously brings a diverse set of interests and skills to the MDZ here. Again, I'm really looking forward to working with Nikki. I think today she's going to focus more on her doctoral research, but hopefully not too distant future, we'll be updating on some of the tuco plans that we have and things going on there. I won't take any more of Nikki's time. Her title is here. Today she'll be talking about the role of reward and dopamine signaling in prairie vole and cortisogaster peer relationships. So thanks, Nikki. Maybe someone hit the lights back there down a little bit. Thanks.
All right. Thank you so much for that introduction, Eileen. Hi, it's really nice to be here today. So today I will be talking largely about prairie voles, so largely about my dissertation research, with a very, very tiny preview at the end of what I'm going to be doing with Eileen with the tucos. So if you want to hear more about that, I am giving an IB seminar, the Postdoc on Parade talk in a few weeks. So I plan to focus largely on ongoing and future work in that talk. So this talk will be largely past work. Okay. So first, here's an overview of what I want to go over with you today. I like to always start with a little bit of summarizing of some conceptual framework that I use to approach my research. And this is the exact crowd where I don't need to convince you that these things are important, but I will note that there are many crowds where I do have to convince people. So first, of course, is that there is huge diversity of life. There's huge diversity in social behavior and the mechanisms that mediate those social behaviors. And so it's really important to study many diverse species. So although most of my work thus far has been in prairie voles, my goal for current and future work is to study increasingly more non-model organisms. So this is where we get into, you're the crowd who is already convinced, right? But there are many, many life science fields, including, like all of biomedical science and neuroscience, where most studies are still being conducted in rats and mice. And so diversifying models in those fields is really, really important and still quite novel of a take. Okay. But of course, not all species are going to exhibit the behaviors that you're interested in or be ideal for the thing that you're interested in, right? So still there has to be some choosing of particular species to study particular things. That said, despite huge diversity across species, there's also evidence for conservation of mechanisms, at least some mechanisms in some cases. And so that means that we can maybe translate across species, which is really important, especially if we care about things like human health. And so the more species that we study, the more we understand what is translatable and when, right? So this is an example I always show of regions involved in the reward and social behavior networks that are maybe homologous across really different taxonomic groups. So amphibians, teleosts, mammals, and so on. Still, even within closely related species, we sometimes do see differences in social behavior and differences in the mechanisms that underlie it. So again, returning to the fact that we need to look at a lot of species. Oh, I wanted to note real quick, please ask questions if you have them throughout. I do prefer that. I don't know if people have a style preference, but I prefer more of a discussion than like me, saying things to you for 40 minutes. So please interrupt me I'm very bad at multitasking though, so do what you need to do to get my attention. Okay, so very broadly, I'm interested in the question, why be social? How be social? And so I want to note that there's obviously a lot of variation in sociality, even in mammals, which I am interested in. All mammals are at least a little bit social, however, because they show at least a little bit of maternal behavior. So that's not true of all animals. But there's variation in the number of individuals that an animal will interact with, how they're related, and how they interact, and so on. So different social structures. So most loosely, interaction between conspecifics, two more individuals of the same species, is maybe social behavior, right? And by this definition, animals who come together only to mate are the least social species. So I have here the example of a polar bear, again, because they're mammals, they do exhibit maternal behavior, which is pretty darn social. But as adults, they are largely solitary. So they spend most of their lives sort of wandering in really vast home ranges and not overlapping very much with other adults, except to mate, if they can find a mate. So what I'm interested in, however, is something a little bit more specific than this loose interaction. So what I'm interested in is affiliative or pro-social behavior. So behavior that brings individuals together. And so where we have polar bears on the least social side of the spectrum, I put a picture of the BEERY lab during the pandemic toward more social. I'm not claiming that we're the most social, but we are quite social. Humans have really complex social structures. We have long-lasting social bonds with many different kinds of individuals. And so sociality varies across species, across season, development, and sex. And studying social versus non-social, whatever that means, is a really, really important way to understand the mechanisms that underlie sociality. So I like to approach my work with proximate and ultimate questions in mind. So ultimate evolutionary why questions such as, what are the costs and benefits of being social? And proximate mechanistic questions, how questions are often called, such as how is social behavior mediated by neural and hormonal mechanisms? And so most of what I have done so far is largely answer proximate questions, but I am interested in starting to approach those ultimate ones and ultimately to perform integrative research. So again, I'll talk about that like a very, very little bit at the end. All right, so social relationships can be broken down into different kinds. This is not necessarily exhaustive, right? But we have pair bonds, so bonds between mates. Parent-offspring bond is pretty self-explanatory. And then peer same-sex relationships, which are essentially friendships. I really don't love to call them that, but they're essentially friendships. And so the first two are reproductive relationships, right? They are to do with reproduction, whereas the third category is non-reproductive. And so a lot of work on social behavior and on the neural mechanisms that underlie social behavior have been done in these reproductive relationships. And so peer relationships are largely understudied, but they are still really important. Okay, so they support group living. And what I mean by that is if you think about any group living species, most of the interactions that an individual will have are going to be with peers, right? That is still a really important part of that social group. So it's not just with mates, with offspring, and with parents. Yeah.
Do we see peer relationships amongst heterosex, so like male-female peer relationships? Do we observe those?
Oh, I don't know that we've studied that. Yeah. Prairie voles are so eager to mate. I'm not sure. Yeah, they're very eager to mate. So like whatever minimum time they're supposed to be together to form a partner preference or to get pregnant, they always do better than that. And by that I mean very short amount of time. So I'm not sure. I will note that prairie voles make all three of these kinds of relationships, which is one of many reasons why I'm very interested in them. All right, so a nice segue into introducing voles, which a lot of you probably have already heard a lot about. But it's never a bad thing to look at more pictures of cute voles. So very briefly, these are prairie voles. These are prairie voles huddling. And they've been largely studied for their social monogamy. So males and females will come together and form pair bonds and mate kind of for life and provide biparental care. So this is really quite interesting, right? Most mammals are not socially monogamous. So whereas, for example, most birds are, fewer than 5% of mammals are socially monogamous. So we're really interested in them as a model for maybe what our own social structure looks like. A lot of work has been done on prairie voles and oxytocin, lots of other mechanisms as well, but oxytocin has been a favorite. Oxytocin is a peptide hormone produced in the hypothalamus. So when released into peripheral circulation, so into the blood, it has a lot of physiological effects, most to do with reproduction, especially maternal behaviors, so like milk letdown. It has many different central effects, so effects in the brain. And a lot of what we know about oxytocin and these central effects in the brain on social behavior, we know because of prairie voles. So they've been really, really important for this. Oxytocin is also highly conserved evolutionarily. So across very, very different organisms, we see something that looks like oxytocin that is doing something related to reproductive, maternal, or even social behaviors. So I won't be actually talking about oxytocin, but I just wanted to provide some setup for why prairie voles have been studied in the past, why they've been interesting, and eventually I will be looking at oxytocin again in the future. But this work on prairie vole pair bonding and oxytocin has informed our understanding of things like human love, human social bonds, and even instances of human social behavior dysfunction, such as autism spectrum disorder. So this has been a really big deal, basically. But a lot of this, or all of this work thus far, has really been on these pair bonds, right? And these pair bonds might not actually translate to all kinds of social bonds, especially in humans, right? So again, this idea that peer relationships are important in prairie voles and humans, and we need to study them to really understand social behavior as a whole. All right, this is the famous partner preference test. It was developed in prairie vole pair bonding work, and it's very simple, but like very, very central to a lot of the prairie vole literature. And so what we have here is an apparatus where the focal vole pictured here can move freely for 3 hours. And the familiar animal is tethered to one end and an unfamiliar animal is tethered to the other. And so the behavior we're most interested in is huddling, which you can see here. So basically when they when they have a lot of their body overlapping in some way is huddling. Yeah. And so what we're looking for is huddling for a partner over a stranger. And it turns out that both the selectivity and this huddling is pretty unique to voles. So rats and mice, which I've already said, have been the favorite model in a lot of different fields, including behavioral neuroscience so far, are social in that they live in large social groups and are gregarious, right? But voles exhibit these selective social preferences, which I'll talk about a little bit more, that are ideal for models of social bonds, right? So choosing one individual over another, not just like generally being nice to everybody. So here we have results from a comparative partner preference test. On the y-axis, we have time in each chamber. So the reason we're not looking at time huddling here is it turns out mice don't really huddle. So we couldn't really compare that. But we can compare time in each chamber in the partner preference test. And what we find is that mice actually spend more time in the neutral chamber where there are no social stimuli than in either of the social stimulus chambers, whereas prairie voles are spending way more time in the partner chamber. So this figure is essentially showing the same thing, but with a couple more species thrown in. And so what we see is mice and rats, we don't really huddle. Degus are huddling, but not super selectively. Metavoles are a little bit selective, but here we have prairie voles, which are highly selective. So a huge amount of partner huddling and virtually no stranger huddling. And we see this over and over again. And time again in mates and in peers, we've done a bunch of things to try to block this behavior, and it's very difficult to do so. They really, really, really want to form these selective preferences. And so related to that selectivity is selective aggression. So one of the things I was really interested in during my PhD is the idea of what it means to be selective, right? And how that's related to aggression. So I think it's really easy to think like, oh, prairie voles are these cute little rodents that we study because they love each other, right? And so they must be super nice. But they're actually, because of the selective aggression, pretty not fun. Well, I love them, right? But they are difficult animals to work with, and they are quite aggressive toward individuals outside of their in-group. And this is a really important part of their social behavior. So again, we see here mice comparatively less aggressive, and their aggression is not super targeted. And then prairie voles on the extreme end with high aggression and very targeted aggression. So aggression at unfamiliar individuals. So again, this is a really good model of a social bond, even a social bond in humans. Right? So social bonds that we form, we might not actively be fighting people, hopefully, right? But we have our in-group that we share resources with, that we form these affiliated bonds with, and then there's the out-group. Okay, so a lot of what we know about mechanisms underlying peer relationships has been done in meadow voles and done by Annalise here. And so what we know is that meadow voles show seasonal differences in their social behavior. So in the winter, they form communal huddling groups and are fairly tolerant of one another, whereas in the summer, especially females are very, very territorial and aggressive, and so relatively solitary. And this can be simulated in the lab by changes in day length. So short day length is winter, they're pretty social. Long day length is summer, they're pretty not social. So just to recap, prairie voles and meadow voles. Prairie voles are socially monogamous, and that's largely why they've been studied. Meadow voles have been studied in their own right as well, but in that early prairie vole work, they were studied a lot as a contrast to prairie voles. So they were closely related, but they were not socially monogamous. And then related to that, prairie voles show biparental care and meadow voles do not, but both show selective peer relationships. And so this gives us a really ideal opportunity to ask comparative questions. So are the mechanisms underlying relationships in voles species or relationship type specific? Do we expect peer relationships in prairie voles to look more like meadow vole peer relationships or to look more like prairie vole pair bonds? And so this essentially was my dissertation research. Okay. Any questions before I move on? Great. So the first project I conducted in Annalise's lab, so long ago now, it's kind of scary, lay the behavioral foundations for everything that came after. So very basic question, what are prairie vole peer relationships like? There was one study before this that specifically looked at peer relationships in prairie voles, and that's about it. There are some peripheral studies that used prairie vole peers to look at other things, but one that directly assessed it. So we didn't know a lot. So what do these peer relationships look like? How do they compare to meadow voles? So I want to note that there are going to be some pictures of undergrads in the upper right-hand corner of many of my slides. These are not necessarily all the undergrads who worked on a project, and some of these undergrads worked on multiple projects, but here's some of the big ones that I wanted to thank and include in my slides. So I compared across sex, day length, and species using a suite of behavioral tests. The partner preference test I already described to you. The social interaction test, which is shown here in 2, is essentially a test of aggression. So we put the focal animal into a novel cage, let them habituate, and then throw in a novel animal and see how they interact. And so in prairie voles, how they interact is largely aggressively, which is why it is a test of aggression. The open field and light dark box tests have essentially the same logic. So rodents are going to want to spend time on the edges of things and in the dark because that's where they're less visible to predators. And so these tests are assessing exploratory behavior and anxiety-like behavior. So the more time spent in the center and the less time it takes to go out into the center, the more exploratory and the less anxious. And then the more time spent in the light and the less time it takes to go into the light is a similar thing. And so the reason we used all of these tests is that anxiety, aggression, and sociality are all interrelated. So sociality may just be the decrease in aggression toward conspecifics and the decrease in anxiety so that you can approach a conspecific. Yeah.
Did you assess whether the strangers were relatives, like siblings or cousins or something, or totally unrelated?
Yeah, we always chose unrelated strangers.
Okay.
Yeah. Yeah, that's a really good question. And a related piece of information is that the peers themselves were sometimes siblings and sometimes not, and we didn't find that there was any behavioral difference. Okay, so what we find is really robust partner preference in prairie voles for peers. So on the y-axis, we have huddling time in the partner preference test. We see that there's partner preference in short day meadow vole females, which we already knew, right? So we knew that short day meadow vole females are the ones who are social. And so what we're confirming here is this partner preference in the long day prairie females. And we see really, really selective preference for a partner. I will note the reason that I'm showing you long day prairie females versus short day meadow females, that may seem a little weird, is again, short day is when meadow voles are social and thus when they have largely been studied in the lab, whereas most of the work in prairie vole pair bonding has been in long day animals, right, in breeding animals. So we wanted to be able to directly compare. We also didn't find day length differences in prairie voles, for what it's worth.
I have a question. So
Yeah.
the short day is in the wild is associated with colder temperatures,
Yes.
and a lot of mammals, come together for sort of group thermal regulation when it's colder.
Mhm.
Can you induce that with changes in temperature in the lab, or is it really about daylight?
I think it's really about day length. I don't think temperature alone can do it, but one of the factors for the communal huddling is thermal regulation. Yeah, but it's not a necessary, or it's not sufficient. All right. And so what we see here in 2 is robust partner preference for a new partner. So in the first one on the left, what we're seeing is partner preference for a partner that they've had since weaning. And so here, instead, what we're doing is in adulthood, we're taking them apart from that original partner they've had since weaning and giving them a new partner in adulthood. And so they are also still able to form highly selective bonds for these new partners. And this is really important because if we're interested in the formation of new relationships in adulthood, we have to be able to make them make new relationships in adulthood. All right, and we also see some pretty distinct species differences in the anxiety and aggression tests that correspond pretty well with the social behavior. So in the top, we see evidence that prairie voles are more aggressive, but also show more social exploration than meadow voles. At first, we thought that was a little bit weird, but probably what's happening is prairie voles are less inhibited. So they are approaching individuals, and it might not always end up in like nice behavior, right? They're approaching individuals more and then being aggressive toward them. And then related to that, prairie voles are also less anxious than meadow voles. So they're less anxious, which is allowing them to approach and then be aggressive. All right, so like meadow voles, prairie voles display robust peer partner preferences. But like prairie vole pair bonds, prairie vole peer relationships are highly selective. So I want to note again, meadow vole peer relationships are also selective, but it looks like maybe prairie vole ones are even more selective. Yeah.
Does the aggression in the prairie voles go from being aggressive for the strangers to wanting that to be like a social interaction?
Oh, that's a really, really good question. So those social interaction tests were like 10 minutes long, and I will tell you 10 minutes is not enough time. When we give prairie voles new partners in adulthood, they are often a little bit aggressive at the beginning. And it usually dies down, I would say, within a half hour. This hasn't been precisely looked at, so that's why I'm hesitating so much. Yeah, they're pretty, I mean, there are some occasions where they never die down and we take them apart and we exclude them from the test, but largely prairie voles are like very ready to go for mates and peers. Yeah. But that doesn't mean they're friendly. Something I always like to know. Okay. All right, I thought that I could wear this jacket, but I really can't. Okay, so the second project of my dissertation used pharmacological manipulations of dopamine to ask what is the role of reward and dopamine signaling in variable peer relationships. So dopamine is a neurotransmitter involved in a ton of different things, some of which include reward and motivation, learning, activity, and movement. And so it's probably no surprise that the dopaminergic reward pathway is really important for variable pair bonds. So they are essential to the formation and maintenance of these bonds. So here we have a bachelor VOLE, meaning his new female. And during this initial formation of a new bond, dopamine D2 receptors are really, really important. And then once they've already bonded, so the period of time that we now call maintenance, dopamine D1 receptors are really important. So dopamine is super important both for the initial formation of new mate preferences, but also the persistence of these mate preferences. And so an early study in variable mates used pharmacology to manipulate these partner preferences. So they used a general dopamine antagonist haloperidol before pairing with a new opposite sex partner and a cohabitation time that should be sufficient to form that preference. They also used a dopamine agonist before formation with a new partner and a cohabitation time that's usually insufficient. And so what they find is that dopamine seems to be necessary and sufficient for partner preference formation in variable mates. And so what that means is haloperidol is blocking partner preference formation even after 24 hours, which is enough time usually. And apomorphine is inducing partner preference formation in 6 hours, which is usually not enough time. Sorry, the cursor is just going wildly. I feel like I'm pointing at things, but I don't think I am. So what about medable peers? Annalise looked at this and she found evidence that haloperidol is not blocking partner preference formation in medable peers, so medable females for females. And so then the natural question is, what about variable peers? And so again, do we expect variable peers to look more like medable peers, or do we expect variable peers to look more like variable mates? So what we find is no evidence that dopamine is necessary. So we used multiple doses of haloperidol, some of them very, very high. And at no dose is partner preference blocked in variable females for females. We do, however, find some evidence that dopamine is sufficient. So here is the control vehicle injection. We see that there is no preference for the partner over the stranger. Actually, decently high stranger huddling and a lot of variability in stranger huddling, but with 1 dose of apomorphine, there is partner preference. So significant partner huddling over stranger huddling. Yeah.
Which species were you doing the injection into?
These are variables. Yeah. These are just variables. Yeah. Yeah. So variable females given new female partners. All right. Okay, so dopamine is sufficient, question mark. This is interesting, but it is difficult to really think about in a meaningful way without its necessity as well. Usually those go hand in hand. And so it's just an example of us having to exercise caution when using dopamine or opioid agonists, like feel-good drugs, to reinforce or condition behaviors, right? So this might not be showing like an endogenous natural process, but it's still interesting. Okay, and aside from the pharmacology, we do still find evidence that peers are rewarding. So dopamine and reward are often talked about like they're the same thing, but they are of course not. There are reward mechanisms that aren't dopamine, and dopamine does other things besides mediate reward. So we find behavioral reward in variable peers. And so this is the socially conditioned place preference test. Essentially, we present variables with 2 novel beddings. One, the tech fresh is super fluffy and is highly preferred to the corn cob, which is really grainy and uncomfortable. And so this is on purpose. So we then condition the corn cob to be associated with social housing, and then the tech fresh to be associated with social isolation. So preferred bedding with social isolation, dispreferred bedding with social housing. And so after initial pretest to make sure that they are showing the preference that we expect, they then go through 6 days of alternating bedding and social housing types before a post-test at the end. And what we see is a really significant increase in the amount of time spent on this conditioned stimulus between pre and post test. And so this is evidence that prairie voles are conditioning toward the social housing, and thus evidence that they are being rewarded by being with a peer. So we have other evidence of behavior reward as well. I was less involved in this project, but it is very, very cool. And you've probably seen it before. But basically, we had animals learn how to press a lever to gain access to a mate or a peer. And so this is all female focals. And it looks like in females, they will press significantly more for a mate or a peer than a stranger. So this effect is stronger in the mates, but it's still here in the peers, right? So partner over stranger pressing, female prairie voles will press a lot more for a peer partner than for a same-sex stranger. And so again, this is showing reward because they are motivated to access that female peer, right? Not quite as motivated, but still quite motivated to work to gain access. So like medieval peer relationships and unlike prairie vole pair bonds, prairie vole peer relationships do not rely on dopamine. Still, there is a lot of evidence for behavioral reward. And so probably it looks like for prairie voles, we have prairie vole mates as like the most rewarding, most highly reinforced, most motivating, followed by prairie vole peers and then mate peers, or medieval peers, sorry, sort of at the lower end of reward and motivation. Yeah. Okay. So the final project I will be talking to you about today looked at social environment and structural changes in dopamine signaling. So how does social environment affect dopamine plasticity? So what we know from the prairie vole pair bonding literature is here. You can see it here. Upregulation of D1 type receptors, so dopamine D1 receptor, is really important in animals that are maintaining their bond. So we say that paired for 2 weeks is when they're maintaining their bond. So D1 is increased in the nucleus accumbens after pair bond maintenance. And so we wanted to look at this in prairie vole peers as well. So this allows us to look at endogenous dopamine regulation and structural long-term changes in dopamine function, which pharmacological manipulations can't do. Okay? And there's still this question of high selectivity. So prairie vole peers are highly, highly selective, and prairie vole mates are also highly, highly selective in a way that medieval peers aren't quite. So maybe this is the mechanism that's making them look more like prairie vole mates. I won't go over this in any detail, but essentially we had many different social housing groups. So we had males and females who were pair housed with a same-sex partner since weaning. These were the control animals. We had males and females who were solo housed for a long period of time. And then females who were given new partners in adulthood and allowed to maintain that new bond. And then finally, we put groups of females together since weaning to look at what group living animals and groups actually look like. And so after these housing paradigms, they went through social interaction tests, which we already talked about, and partner preference tests. And then we took their brains, sliced them very thinly on a cryostat, mounted them to slides, put them through a series of washes before using a selective radio-labeled ligand to look at the receptor of interest, in this case, dopamine D1 receptors. Yep. And then what you get is, after exposing slides to film and developing them, you get this. Okay. So let's look at this one first. So we don't find a difference in control versus repaired voles, which is what we did find, or what other people found in prairie vole pair bonds, right? So again, in the mates, when they're given new mate partners and then allowed to maintain that bond for 2 weeks, there's an increase in dopamine D1 receptors in the nucleus accumbens. So we do not see that same upregulation. What we do see, however, is upregulation of dopamine D1 receptors in the nucleus accumbens in isolate animals compared to control animals. So at first we found this a little bit confusing, but you'll see in a little bit that it actually paints a really nice and consistent picture. And it looks like this is fairly consistent with work in other rodents. So novel finding in voles, and not the exact same thing has been found in other rodents, but in mice, rats, and mandarin voles, we see that early social deprivation, so social isolation early in life, is also associated with upregulated D1 in the nucleus accumbens. So we'll come back to that story in a little bit, but I want to note here, this is behavior from the partner preference test, again, huddling time on the y-axis, and we see that every single group that had a partner showed significant partner over stranger huddling. And this is pretty consistent with what we found in a lot of other experiments previously. So it looks like manipulation of social environment, even manipulation of different pharmacology is really not blocking this preference. This is a very, very strong preference that's very difficult to manipulate. And then here we have behavior in the social interaction test. On the y-axis, latency, the first attack. So the shorter the latency, the more aggressive the animal, right? And what we find is that isolate females are more aggressive than isolate males. So this is interesting. Prairie vole males in every context are usually more aggressive. So in a mate context, they're the ones who are largely doing the mate guarding and the ones who are largely territorial. And even in a peer context, we actually had to stop pairing them in adulthood, which is why they are only in the control and isolate groups, because they were so much more aggressive than the females. So in that initial project where we laid the behavioral foundations, we were pairing males in adulthood because we wanted to see if they could, and we were planning to pursue that as well. They were really, really aggressive. And so maybe even like half the time, I don't remember the exact numbers, we had to take them apart. They were not going to be friends. So there is some evidence that we are seeing this because females might be more sensitive to social isolation than males. So there are experiments that have looked specifically at social isolation and how it can induce depressive and anxiety-like behaviors in prairie voles that have found this. So hormonally and behaviorally, isolate females seem to react more strongly to the isolation than isolate males. So this might be causing the increased aggression. Okay. So we also assessed individual behaviors and correlations between individual behavior and binding. And what we find here is a story that is fairly consistent with what we find right here, which was pretty interesting. So basically in the partner preference test, higher nucleus accumbens D1 binding is positively correlated with more time spent alone in the PPT, so a social behavior or antisocial behavior, sorry. And then again, higher nucleus accumbens D1 binding is positively correlated with more time spent grooming oneself in the social interaction test. So this is another antisocial behavior. So when they're grooming themselves, they're not grooming another animal, they're not doing anything more interesting, right? So D1 binding seems to be associated with more antisocial behaviors. What we also saw is a lot of correlations across the test types, which was pretty interesting, and shows evidence of maybe, something like personality or consistent behavioral phenotype in these voles. And so here's just one example of what we found. So higher huddling partner bouts in the PPT was negatively associated with sniffing frequency in the social interaction test. So the reason this is consistent, right, is the PPT is looking at partner-directed behavior, whereas the social interaction test is looking at stranger-directed behavior. So the more time they spent huddling, or the more number of bouts that they had huddling a partner in the PPT, the less time they spent sniffing a stranger in the social interaction test. So there seems to be an association between the degree of partner selectivity and how interested they are in a stranger. Yeah.
Did you look at D2 binding in the nucleus accumbens?
Yes, we did. Yeah, we didn't really find anything interesting. So D2 binding took a very, 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 very long time to develop, which was a big problem. So basically, like months after Annalise moved and months after I moved away, it was still developing because all of our test developments were like very faint binding. And so what we end up with is very, very low binding against a pretty dark background. And so we didn't find anything really interesting. Yeah, but we did look. Yeah, this paper is actually has been submitted to neurosciences and is under review right now. And I expect that they're going to ask about the D2 binding. So unlike with prairie mates, dopamine D1 receptors are not upregulated after long-term maintenance with a peer. Instead, it looks like isolated voles are showing upregulated nucleus accumbens D1 binding. And also it looks like high D1 binding is correlated with more time spent alone and less time being social. So these two things together are giving evidence that it's a two-way relationship, right? So high D1 binding seems to be both cause and consequence of reduced social behavior in these animals, which is quite interesting. And so altogether, all of these projects provide strong evidence that reproductive bonds are mediated differently from non-reproductive ones. So in everything that I've looked at, this has been the case. And this is more evidence that prairie peer relationships and peer relationships in general need to be assessed in their own right, because the mechanisms that mediate them are likely to be quite distinct. Okay. And so in ongoing and future work, I hope to move toward more naturalistic study of behavior. So here is a picture that many of you are probably quite familiar with. So this is a live trap, and this is a vole runway. So we occasionally outbreed with wild populations in the vole world because we care about genetic diversity and because we care about natural behaviors. And so this is sort of just the beginning of my path towards studying increasingly more naturalistic behavior, right? So entrenched in this field that is doing a lot of lab work, but does still care about this. I started to move into actually studying group living animals in groups toward the end of my PhD. And then we'll actually start studying animals in semi-natural enclosures and in the field in the future. So this project I started toward the end of my PhD. It is not part of my dissertation. It was sort of an extra. And I continued it in my lab at Colgate. So I was an instructor at Colgate. It was largely a teaching postdoc, but I also did run my own lab. So I was essentially principal investigator of my own lab with 8 undergrads for a year. And this is what we worked on. So basically we use a very mild behavioral stressor. Voles float and are very good swimmers. So very brief amount of time in a swim test, gave them new partners and then assessed their partner preference. We also did this in groups of animals. So animals that have been together since weaning, these are same-sex female prairie voles. Swim tests, and then how are they behaving afterwards? So there's some evidence, this is still actively being scored and analyzed, but there's some evidence that stress may be impairing relationships between peers and prairie voles, just like they are in prairie vole females in pair bonds and meadow females in peer relationships, which is pretty interesting. And there's also some evidence that stress may be decreasing affiliation in these group living animals. So in this pre-stress test, what we're seeing is most of the animals spending most of their time group huddling. They really like to huddle. But it looks like after this very, very mild stressor, they actually spend a lot of time apart, which you don't really see in both. So again, this is very preliminary, but it looks like stress is altering the ability to form partner preferences and also the dynamics within a group in prairie vole females. Yeah. At Colgate, we also followed up with some tests where instead of stressing all of the individuals out, we only stressed out a subset, just because it should provide a really good opportunity to look at things like constellation behavior and social buffering, which prairie voles do show for each other. Yeah. So like I said, this is actively still being scored and analyzed. And I actually have a few volunteers from my lab at Colgate who are continuing to work with me on this project, which is pretty exciting. Okay, so very brief preview of the TUCO stuff I will talk about more in a few weeks. But with Eileen, I'm accomplishing this goal of moving increasingly toward more naturalistic study of diverse rodent species. So here are tucos. I think you've probably all seen pictures of them. They are incredibly cute and soft and so much more docile than the prairie voles ever were. And then here is not the actual enclosures we'll be using. We don't have a picture of them, but enclosures at the Berkeley Field Station that Michael Nachman provided for me a long time ago. I don't know if you even remember that I emailed you, but And so very briefly, we'll be looking at neural and genetic predictors of dispersal in colonial tucos, both in the lab and in semi-natural enclosures at the field station. So dispersal is really important in the lives of many, many animals. It affects social organization, population diversity. It affects so many different biological levels, but we actually know basically nothing about the neural mechanisms that is mediating dispersal. And so I will be looking at that to see if we can find predictors of dispersing versus non-dispersing individuals. And it also is related to social behavior, which is where my background is, because in a lot of species, dispersal is marking the shift from more to less social. So a lot of species are fairly solitary as adults, but when they're young, they're quite social. They're with their litter mates, with their mother, and at some point they leave. So there's this big shift in social behavior. Similarly, Annalise and I have been spending the last few years laying foundational work to really integrate field work with lab techniques, which is, of course, my ultimate goal in Belding's ground squirrels. So here she is pictured holding a Belding's ground squirrel. They are wild animals, so you have to hold them pretty tight. And this is our beautiful field site right outside of Yosemite. So gorgeous. And to be frank, this is the only field work that I have done. So I know that you all have gone to really exotic locales. This is where I've gone, and it was very lovely. And this is in collaboration with Scott Nunes at the University of San Francisco. So in these animals, I will also be looking at dispersal and behavioral neural hormonal correlates of dispersal. And it's really important to study animals in the context in which their brains and behavior have actually evolved, right? So really important to look at wild animals. Okay. And so with that, I'd like to thank the BEERY Lab, since that's where I conducted almost all of the work I presented today. BEERY Lab, some pictures at the top. And then also my COLGATE VOLE lab, pictured here at the bottom. So it was a very interesting experience, but it was cool to basically be PI of my own lab. And these were my undergrads there. Yeah. So thank you.
I'll hit the lights. I know Annalise was taking questions along the Annalise.
I was trying not to.
Yeah. That was, I don't know what that slip was. I think you have to send Annalise an email right after this.
I'm mini her, it's
That's what I'm
okay.
thinking. But anyway, Nikki's been taking questions along the way, but I'm sure she's happy to answer questions if people have questions now. So, yeah, Carol.
Just related to that same question I asked before about, siblings and the strangers there.
Mhm.
So in the wild, with the prairie voles, are there peer friends that they do hang out with that aren't a mate? Are they related to them usually, or are they sort of random or?
Yeah, so there's some evidence that there are peer-peer interactions in the wild, but to be honest with you, it is less relevant to prairie vole social systems than meadow vole ones.
Okay.
So it's a less extreme shift, but prairie voles also form communal huddling groups in the winter. And generally these consist of related individuals, but sometimes also unrelated ones. Yeah.
Other questions?
Yeah.
For your swim test, how do you do a control for that?
So we just didn't stress them at all. We just handled them and didn't plop them into water. Yeah. So it's worth noting, well, maybe none of you actually thought of this, but there is like a more severe version of this test that is trying to induce depressive-like behaviors in animals. So this is like a very mild version of that. Very brief in room temperature water. They just for a little bit and then we take them out. Yeah. Yeah.
Okay.
On a related note, is stress related to the D1 receptor?
Oh, I'm not sure. Yeah. So it seems like it might be, right? So I don't know that it's directly been studied in even prairie vole pair bonds, right? But the fact that we found things in dopamine, D1, and in social isolation, which is stressful for prairie voles, it is stressful for them, seems to suggest that there is a relationship. So from the prairie vole pair bonding literature, people have separately found that a lot of things, right? But stress and court and oxytocin and dopamine and opioids are all really important for prairie vole pair bonds. And they have found really direct relationships between dopamine, oxytocin, and opioids, but I don't think stress and cork. Hopefully I'm not wrong about that. Yeah.
This is a naive question. How long do D1 receptors last once they're assembled on the membrane? I assume it's on a, you know, on the surface of the cell.
Yeah.
And it sounds as if it's, you know, your assays see variation in the amount that's, so I'm just wondering how plastic that is. What's the rate of turnover once receptors are built on the cell surface? Is it something that changes on a scale of days, or is it seconds or minutes?
Yeah, I do not know. Therefore, that must not be a naive question because I don't know. But it is probably on the scale of days because we have found plasticity within 2 weeks, right? So it's at least at that scale, which to me is a pretty moderate scale. So it's not really fast like seconds, but it's not like months. That wasn't really an answer, but that's a good question.
So I'll ask it completely different, not for something completely different. If the take home is that the type of social bond is more important than, say, being a prairie vole versus being a meadow vole, and knowing that a lot of mammalian social systems are built on female-female peer relationships, where would you go next? Meaning, what system would you want to look at next to look at, say, female-female peer relationships?
Do you want me to say tooth goes?
No.
What you were,
Well, I just thought about expanding that work, you know, based on what you know about social relationships with mammals, thinking also from a phylogenetic perspective. And no, tupos would not be a good choice because they don't form particularly strong bonds.
Yeah. So that is one line I'm interested in the buildings ground squirrels. So like a lot of animals, largely males disperse, sometimes females do, but a lot less so. And so females will stay at the natal nest, and then sometimes even show, you know, cooperative breeding together. And so you're likely to see maybe peer relationships there. A lot of them are related, but it's still, I think, worth looking at. I also sort of proposed to do work on tree squirrels while I was at Colgate. So I got the permits for it and everything and didn't end up doing it. But what we find, I can't remember the details, that's why I'm speaking very slowly. But what we find is that eastern gray squirrels are really easy to find, obviously, in the Northeast. And then fox squirrels, a little less so, but they also do overlap in range. And we see pretty distinct differences in how social females are in gray squirrels and fox squirrels. So if I remember correctly, fox squirrels, females are less social with one another, whereas gray squirrels, there's evidence that maybe they're more social compared to fox squirrels. And this also interested in me because it looked like the social systems were very, very distinct by sex. So males had their own social system that was very distinct from females in both of these species. So that's something I've thought about. Yeah.
Well, Brett, I was gonna say, you might just want to talk to Chris Conroy too about what other roles are doing in terms of if there are other goals.
Yeah.
Social female relations.
Yeah, I've talked to him a little bit about some like really cool big voles in the middle of the desert.
Yeah.
That I'd like to hear more about. Yeah.
But I think we'll let people get on with their day. Let's a round of applause to thank Nikki.
Someone asked a question in the chat. Oh, I can answer it. Okay. Can you talk about the virtues and pitfalls of applying this kind of research you do to human social relationships? After all, that seems to be part of the research hook. Yeah, so I mentioned it, but it's like not a big part of the research hook for me. I personally do not care about humans, which I probably should stop saying for the rest of my career. So one virtue, of course, is that prairie voles are showing a social system that is similar to what we think that we show, but are a rodent that we can actually manipulate and assess. But obviously the major pitfall is that they are very different from us, and they are a lot less plastic and a lot less complex in their social behavior than we are. So I think the major pitfall, right, is that a lot of the work on prairie vole pair bonding has been translated to humans in a really simplistic way that's probably not useful, right? So the idea of oxytocin as a love hormone is a really good example of that. Oxytocin is not a love hormone. There are many situations where oxytocin is actually increasing antisocial behavior. Yeah, hopefully that answers your question. Person in the chat. Hi.
I just wanted to introduce
Oh, hi.
myself. I'm Jim Patton. I'm
Oh,
a
hi.
longtime curator